Emasculation, removal of anthers. Emasculation performed in bisexual plants. Okay. Genes. Mendel's called it factors. Alleles. Genes which code for a pair of contrasting traits are known as alleles. Alleles, they are slightly different forms of the same gene. That is term homozygous, heterozygous. Homozygous capital T, small t, heterozygous capital T, small t. That is factor, a pair of factor that can be a dominant factor and can be recessive factor. So in Mendel's laws of inheritance, in law of dominance, one factor is dominant, that is dominant factor over the recessive. Okay. Now monohybrid cross. Okay. Monohybrid cross. Since the capital T small t plant is heterozygous for genes controlling one character height, it is a monohybrid, and the cross between capital T small t is a monohybrid cross. Monohybrid, a single hybrid, that is capital T small t, and the cross between the capital T and small t is the result of capital T small t. Is the resultant is capital T small t. So monohybrid and the cross is monohybrid cross. The result of monohybrid cross, the phenotypic ratio is tall is to dwarf, that is 3:1. The genotypic ratio is 1:2:1. Capital T, small t, right? Now we use the term Punnett square is used to understand a typical monohybrid cross conducted by Mendel between true breeding tall plants and true breeding dwarf plants. Mendel selected 14 pairs of true breeding plants, right? Plants. Let's see. So 3:1, 1:2:1. Okay, Punnett square. You see. So Punnett square developed by a British geneticist Reginald C. Punnett. It is the graphical representation to calculate the probability of all possible genotypes of offspring.